This is the review of the digestive system. As with the respiratory system, we're going to start with the main ideas, and you need to make sure you have these really well understood, and then we'll move into the supporting information which will help you remember and apply the main ideas. So the first idea is that the digestive system's main function is the intake of nutrients. You knew this. You eat, that's ingestion. You digest, that is, you chemically break down the food into particles that are small enough for you to absorb. The organs of the digestive system are organized into two categories. There's the digestive tract, also called the alimentary canal or the gastrointestinal or GI tract, and then there are these accessory organs. The digestive tract starts with the mouth. From the mouth, food continues to the pharynx, starting with the oropharynx and then through the laryngopharynx. From there, air would go to the trachea, but food goes to the esophagus. From there to the stomach, small intestine, and large intestine. Accessory organs include 3 associated with the mouth and 3 associated with the duodenum. Accessory organs associated with the mouth include the teeth, the tongue, and the salivary glands, and those associated with the duodenum are the pancreas, liver, and gallbladder. There are a number of processes associated with digestion. I've listed the major categories here. Salivation, the production and secretion of saliva, mastication, which is chewing, deglutition, which is swallowing, and peristalsis, which is the smooth muscle contractions that propel food starting at the esophagus and going all the way through to the other end. There are a number of hormones and autonomic nerves that regulate digestive function and appetite. There are enzymes associated with digestion, and these enzymes are secreted into the mouth, the stomach, and the small intestine. Remembering that the major function is intake of nutrients, it's important to know what the major nutrient groups are, and that is carbohydrates, proteins, and fats. These must be broken down to their smallest units. That would be monosaccharides for the carbohydrates, amino acids for the proteins, and fatty acids for the fats. Breaking them down to their smallest units allows them to be absorbed across the intestinal epithelium. Once they're absorbed, we have to get those nutrients to their final destination in the body. Water-soluble nutrients are carried directly to the liver, but lipid-soluble nutrients have to be packaged into a particle called a lipoprotein, and these are absorbed through the lymph and dispersed throughout the body before arriving at the liver. Finally, waste products must be removed from the body after being processed in the colon and rectum. Let's move on and get the supporting information. We'll start with functional anatomy, and in the category of functional anatomy comes everything that relates the structure to the function. So in this section, we'll spend a lot of time looking at the gross anatomy and the microscopic anatomy, and we'll look at the physiology of each part as we move from the mouth through to the anus. Starting with the mouth, pharynx, and esophagus are the parts of the digestive tract above the diaphragm. Now, you're familiar with the structure of the mouth. You know that salivary glands deliver saliva into the mouth. There are a number of salivary glands, but there are 3 major glands that we learn the names of. The parotid salivary glands are the largest. They're called parotid because they sit next to the ear, so they sit on your cheek just anterior to the ear. The submandibular glands are sitting below the mandible. They're also called submaxillary because they're also below the maxilla bones. And of course the sublingual is under the tongue. Salivation is controlled through a reflex center in the medulla. You salivate when you stimulate the salivation center, and usually that's a positive thing such as seeing, tasting, smelling, or the tactile sensation of food in your mouth. but it can also be stimulated in a negative sense by the vomiting reflex center. Mastication is the proper term for chewing, and it's a combination of voluntary and reflexive motions. These reflex motions are things you don't have to think about. They're just little programs, motor programs in the brainstem. And it involves the skeletal muscle of the jaw for sure, but also the tongue movements, and these are part of the reflex actions. You generally don't think about how you move your tongue when you're chewing. You just learned from experience not to bite your tongue when you chew. Teeth have two major parts. The upper part is the crown and the lower part is the root. The base material on the interior of a tooth is called dentin. Dentin contains the nerve and the blood supply that comes up through the root, and both the root and the crown have a sealed covering. The crown is covered with enamel, which is the hardest substance in the body, and the root is covered in cementum. Swallowing, or deglutition, occurs in three phases, and these phases are controlled by the swallowing reflex center in the medulla. The first phase, the buccal phase, is voluntary. You compress the food into a bolus, you push it against the back of the throat. The soft palate pushes back against the throat to seal the nasopharynx, and that triggers the second phase, the pharyngeal phase, which involves contraction of the skeletal muscle in the throat, and the skeletal muscle in the throat is arranged as a set of circular muscles called pharyngeal constrictor muscles, and they squeeze that bolus of food through the pharynx. At the same time, the larynx will rise up, and we move into the third phase, the esophageal phase. We have to get past the epiglottis, which is going to fold down to cover the glottis, the opening into the larynx, to prevent food or liquid from going into the trachea. Once we're in the esophagus, we hit smooth muscle, and we encounter the first of peristaltic waves. Peristalsis is a specific set of muscle contractions that involves the circular muscle constricting and the longitudinal muscle dilating the esophagus in waves, so that the food is constricted above and the tract is dilated below. This opens the sphincter, and into the stomach the bolus will go. The sphincter will close shut and stay shut. As we look at the esophagus, we encounter the layers of a mucous membrane. The outer layer of the esophagus is the serous membranes of the parietal pleura, because the esophagus is found in the mediastinum, which is in the middle of the thorax. The lining of the mouth, the oropharynx, the laryngopharynx, and the esophagus is all stratified squamous epithelium, and that stratified squamous epithelium protects the living tissue underneath from anything abrasive in what you swallow. The lining of the digestive tract is a mucous membrane or mucosa, and while the type of epithelium will change as we get into the stomach, there's always a loose connective tissue called the lamina propria underneath the surface epithelium. Underneath of the mucosa is a layer of dense connective tissue called the submucosa, and it contains whatever glands are located in that particular part of the tract. In the esophagus, we just call these esophageal glands, and they secrete mucus, which helps lubricate the passage of the bolus of food through the esophagus. The third layer of a mucous membrane is called the muscularis externi, and this is our layers of circular and longitudinal muscle that give us the peristaltic waves. So from stomach through intestine, we still have the mucous membrane. We have the mucosa, which from stomach to intestine will be simple columnar epithelium. We still have the submucosa. We have a very thin layer called the muscularis mucosa. We have that in the esophagus as well. It just doesn't have much to do there. The outermost layer below the diaphragm, though, is called the serosa, and it is the visceral peritoneum on these organs. The muscularis mucosa is a very thin, lightweight layer of smooth muscle cells that when they contract, they kind of fold the mucosa up. And these folds of mucosa are going to be more or less extensive depending on which part of the tract you're in. The digestive tract has an extensive nerve supply. The nerves are organized into two groups. One group supplies the submucosa and stimulates the glands for secretion, and the other group stimulates the contraction of smooth muscle in the tract. So we have a submucosa plexus and we have a myenteric plexus. These are, for the most part, parasympathetic nerves, which can stimulate gland secretion and muscle contraction. Sympathetic nerves are going to be involved in controlling the diameter of the blood vessels of the tract. Okay, so we left off deglutition just as the bolus of food entered the stomach. The stomach is guarded by the cardiac sphincter, which is also called the lower esophageal sphincter or gastroesophageal sphincter. It only opens with each peristaltic wave from the esophagus and then it closes again. This helps prevent gastric reflux of acidic gastric juice into the esophagus. The stomach is divided into four regions. The cardia or cardiac region is simply that small area around the connection to the esophagus. The stomach extends upward above where the esophagus joins it, and that upper extension is called the fundus. The fundus is a pretty quiet area. There are some cells there that secrete one of the hormones that we'll be talking about later. But food can sit here. If the stomach is very full, whatever food you eat last just kind of piles up into the fundus. The main action is in the body of the stomach, and it has a third layer of muscle called the oblique layer. The fourth and final region is the pylorus, and this is a narrow region that, with peristaltic waves, can propel the liquefied food and stomach juice, the chyme, through the pyloric sphincter and into the duodenum. Because the chyme is very acidic as it first enters the duodenal lining is very thick and alkaline mucus. So the duodenal glands secrete this thick alkaline mucus. They are the glands of the submucous layer of the duodenum. The duodenum itself is only about 10 inches long, and it is basically the spot where you mix the acid chyme from the stomach with the digestive secretions of the pancreas and the liver. That would be pancreatic juice and bile. The pH from the stomach is neutralized by these two secretions. Enzymes are added. Everything's kind of mixed in small batches at a time and moves into the jejunum. So fluid doesn't stay in the duodenum for very long. It just moves right along into the jejunum. As it moves through the jejunum, the middle portion of the small intestine, digestion and almost all absorption occurs. And then as it moves into the ileum, we're going to do the very last digestion. We're going to absorb the last of our nutrients, including the bile salts. And the lining of the jejunum and ileum, the mucosa, is very highly folded. When you look at the interior, actually starting in the duodenum, you see a series of spiral folds called plicae. They're kind of like the threads on a screw. They spiral along. That not only increases the surface area, it causes the fluid to swirl as it pushes through. The surface at a microscopic level is thrown into a series of little folds called villi, and each villus has its own blood supply and its own lymph supply. It has its own capillary bed. Remember, these are fenestrated capillaries, so they're very highly absorptive, and its own little lymph capillary called a lacteal. Now when you see lact, that refers to milk. And the reason the lacteals are called that is after a meal they're full of fat and it makes the lymph look very milky. If you look under the microscope at a villus, you see it's a simple columnar epithelium, and these epithelial cells have on their upper surface an abundance of microvilli, forming what we call a brush border. Remember that microvilli are always for absorption. So you have microvilli, you have villi, and you have plicae, which increase the surface area probably about 400 times what it would be without those folds. As the material moves from the last part of the ileum into the large intestine, we just instantly change the name from chyme to fecal material. Fecal material enters at the ileocecal valve. The cecum in humans is quite small, and coming off of it is the appendix, and the appendix is just there apparently more or less to monitor for immune problems. It's full of immune cells. So the cecum is just sort of the entry chamber, and the material moves almost immediately up into the ascending colon. The ascending colon ends as it approaches the liver. It makes a sharp turn to form the transverse colon, which ends at about the level of the spleen, makes another sharp turn to form the descending colon. The descending colon ends about at the level of the hip bone and forms the sigmoid colon, which simply carries the material back to the body's midline. The sigmoid colon ends at the rectum. The colon is involved in absorption of almost all the remaining water, along with ions, mostly sodium and bicarbonate. The intestinal cells of the colon are still simple columnar epithelium, but they're packed with goblet cells, and that mucus secretion helps protect the colon lining from the bacteria that live there. Those bacteria belong there. They're called coliform bacteria. They do us some good. They produce some vitamins, B and K vitamins, that we can absorb, and they're living off the organic waste that we can't absorb, such as plant carbohydrates. From the distal ileum through the colon, there's an abundance of lymphatic nodules, part of the MALT. Remember, that is mucosa-associated lymphoid tissue. And in the ileum, these are quite distinct, and they're called Peyer's patches. They help guard against any bacteria that decide to quit living harmlessly in the lumen of the intestine and try to invade the epithelium. And as I said, the appendix is another lymphoid organ. It helps to create an early warning system right there at the junction between the ileum and the colon. As fecal material moves into the rectum, the rectal wall is stretched, and this triggers a defecation reflex, which means you'll have a strong peristaltic wave to propel feces through the anal canal. The anal canal is only about the last inch of the tract. It just carries fecal material through the skeletal muscle floor of the pelvic cavity. There are two sphincters here. There's an inner and outer sphincter. The inner anal sphincter is smooth muscle and involuntary. The outer skeletal muscle sphincter is voluntary. Okay, looking beyond the tract, there are many membranes that help hold the intestine and other organs in place. These membranes are called the mesenteries, and they grow in the embryo off of the parietal and visceral peritoneum, connecting them to each other. The greater omentum is the biggest of these. It folds down from the greater curvature of the stomach to the rim of the pelvic girdle, and then it folds back up and attaches to the margin of the transverse colon. Ribbons of fat can be deposited in the greater omentum, and this is what people refer to as a beer belly. It's a very superficial layer of fat, not the same as the deep visceral fat that you find within the peritoneal cavity. The second membrane is the lesser omentum, and it connects the lesser curvature of the stomach and the duodenum to the underside of the liver. There is a falciform ligament that divides the liver into right and left lobes and connects it to the anterior body wall. And the mesentery proper is a fan-shaped membrane, attaches to the entire length of the small intestine, has embedded within it the blood vessels to supply the intestine and the lymphatic supply, including many intestinal lymph nodes. There are two regions of mesocolon, the transverse mesocolon and the sigmoid mesocolon. And they attach these regions of the colon to the body wall, to the posterior body wall. The ascending colon and descending colon are retroperitoneal, and you remember that that means they are not in the peritoneal cavity, they actually are embedded in the posterior body wall behind the parietal peritoneum. The pancreas secretes the major enzymes of digestion. These enzymes are produced by pancreatic cells, stored there, and then secreted by those cells. These cells secrete their enzymes into ducts, and that makes these exocrine glands. They're called the pancreatic acini. The ducts contain cells that also produce bicarbonate, and this bicarbonate helps to neutralize stomach acid. Now, the pancreas is a mixed gland, meaning it has some exocrine function, but it also has some endocrine function. The endocrine cells of the pancreas include cells that secrete insulin and glucagon, and the little clusters of endocrine cells in the pancreas are called the pancreatic islets, or islets of Langerhans. The liver is the largest internal organ, and we always associate it with the digestive system, yet it only has one digestive function, and that is the production of bile salts. Bile produced by the liver cells drains from each lobe of the liver through the left and right hepatic ducts. These ducts join together to form the common hepatic duct, and the common hepatic duct connects to the cystic duct. The cystic duct connects to the gallbladder. So if you're not actively secreting bile, bile goes from the lobes of the liver to the right and left hepatic ducts, to the common hepatic duct, to the cystic duct, to the gallbladder for storage. The gallbladder will store and concentrate the bile when you're not eating. When you are eating and you stimulate the gallbladder to contract, bile from the gallbladder moves from the cystic duct to the common bile duct, which then joins with the pancreatic duct to form the duodenal papilla, which empties into the duodenum. Okay, so we've gone through the functional anatomy. We're going to go back through now and try to pick up on some of the digestive activity. So in the mouth, you do begin digestion there. As you mix food with saliva, there's really only one enzyme of note that contributes to digestion of food, and that is an amylase. Amyl means starch, so an amylase is going to break down starch. We refer to this one as salivary amylase because there will be another amylase coming from the pancreas. Amylases can break down polysaccharides, both starch and glycogen. They can't break it down all the way, but they can at least start cutting it down into smaller pieces. Actually, since we don't keep food in our mouths very long, the salivary amylase doesn't usually have much of a role, because as the food gets into the stomach, the acid of the stomach almost immediately starts breaking the food down further, and it will actually digest the enzyme, the amylase enzyme that came in from the mouth. Pepsin breaks down proteins. It doesn't break them down all the way, but it breaks them into fragments. So you start protein digestion in the stomach, and what you end up with before food leaves the stomach is fragments called polypeptides. The stomach pH is very acidic, about 1.8, and that can break down almost anything. So fibers, plant fibers, bone gets dissolved, you name it. The pH is acidic enough to just break down any solid materials. So food won't leave the stomach until it's completely liquefied. As chyme leaves the stomach, it does so in very small amounts. Remember that the pylorus is just letting it dribble through a little at a time. And the chyme will mix with bile from the gallbladder and pancreatic juice. They have a pH of about 8. So they're going to neutralize very quickly the pH of the gastric juice. They're also going to add bile salts, which are going to emulsify the fats, make them mix with water so that they can be processed, and enzymes. And the enzyme that digests fat is called pancreatic lipase or lipase. The pancreas also secretes protein-digesting enzymes, including trypsin, and that should say pancreatic amylase. The gastric glands in the stomach are coiled tubular glands. The neck of each gland is packed with goblet cells, and the deeper part of the gland has 3 different kinds of cells. The chief cells that secrete the enzymes, parietal cells which secrete the acid and a special molecule called intrinsic factor, and G cells which secrete gastrin. Now, gastrin's a hormone. It's not going into the lumen of the stomach, it's going in the opposite direction, into the blood. Let's talk a little bit about the secretion of acid by the parietal cells. The acid secreted is officially hydrochloric acid, but the cells themselves are actually producing carbonic acid. Now, we've met carbonic acid before. We met it in red blood cells where it's making the same thing. It's taking carbon dioxide and water to produce the acid hydrochloric acid. But this acid breaks into 2 parts. It dissociates within the cell. The hydrogen is secreted into the stomach lumen, accompanied by a chloride ion, which is why we say hydrochloric acid is being secreted. The bicarbonate produced from the carbon dioxide is sent into the blood, and that raises the pH in the gastric veins. The only enzyme secreted in an adult stomach is pepsin, and pepsin is secreted in an inactive form called pepsinogen, and the acid itself activates the pepsinogen. Pepsin is unusual because it's an enzyme that can work at such a low pH. Most enzymes are going to be denatured at a pH that high. That special molecule called intrinsic factor is needed to absorb vitamin B12. B12 is a large, complex organic molecule, and this molecule called intrinsic factor literally connects to it, binds to it, covers it, and keeps it from being digested. The B12 won't be absorbed until later in the intestine, but you have to protect it from the stomach acids, and that's what the intrinsic factor is for. A lack of intrinsic factor occurs most commonly in elderly individuals. Their cells simply quit making the intrinsic factor, and they have a deficiency of B12. That condition is called pernicious anemia, and it also results in other problems. Now, in babies, there are a couple of other enzymes that are secreted. Most notable of these is renin, which is an enzyme that coagulates milk solids. Renin is used commercially in making cheese. The first step in making cheese is to take the milk, add renin to it, and that coagulates all the milk solids to make cheese out of them. Pancreatic secretions include the bicarbonate to neutralize the stomach acid and the enzymes that are going to digest all the organic nutrients, and amylase to finish polysaccharide digestion, starch and glycogen, trypsin and other proteases to break down the proteins, lipase to digest the fats or triglycerides to fatty acids, and there are a variety of other enzymes including a DNase and an RNase to break down DNA and RNA to the nucleotides. The bicarbonate is actually produced by the pancreatic ducts, and they use the same process that the parietal cells of the stomach use. They use carbonic anhydrase. They make bicarbonate, but in this case the bicarbonate goes to the lumen of the duct and gets secreted, and the hydrogen ion goes to the blood. So remember, the stomach sent bicarbonate to the blood, the pancreas sends the hydrogen ion to the blood. When pancreatic blood mixes with gastric blood, it neutralizes the change in the pH. Liver cells make bile, and bile has two primary components. The bile pigments, which you remember are just the waste product from hemoglobin, the bilirubin, and if it's not removed continuously, if bile builds up for any reason, that causes jaundice, that yellowing of the skin that's associated with a buildup of the bile pigments. So that's a waste product. The bile salts, on the other hand, have a digestive function. They emulsify the lipids in the small intestine. Remember that lipids don't mix with water very well, so the bile salts have two ends. One end likes lipids, the other end likes water, and the bile salts coat the lipids so that with the mixing actions of the small intestine, you get little droplets of lipid, and each little droplet is coated with bile salts, and that keeps those lipids from recoalescing and keeps the surface area of the lipid droplets large enough for the lipase enzyme to get in and to break down the fats. If you don't produce enough bile salts, then you can't absorb all the fat. You can't digest it, you can't absorb it, it stays in the feces, and so people that have had their gallbladder removed are put on a special diet. They may cheat on that diet, but they'll know about it because they'll have a lot of fat in the feces. Okay, so as a result of pancreatic enzymes, we've digested everything down almost but not quite to the end. The enzymes that finish digestion of proteins and carbohydrates are actually produced by the intestinal cells themselves, and these enzymes are actually attached to the surface of the cell along those microvilli. We call them brush border enzymes. You have dipeptidases to finish the protein digestion down to amino acids. You have a maltase to digest maltose to glucose. You have sucrase to digest sucrose to glucose and fructose. and lactase to digest lactose, which is milk sugar, to glucose and galactose. So you have three different monosaccharides, glucose, galactose, and fructose, that can be absorbed. And once they're absorbed, the glucose, or the sugars and the amino acids, are going to be transported through the epithelial cells and into the blood. So once we get them into the blood, remember where we are. We're in the villus, so we're in those little fenestrated capillaries of the villus. So amino acids and sugars in the blood are going to drain through the blood, the venous drainage of the intestinal veins. This carries them to the superior mesenteric vein, which carries them to the hepatic portal vein, which carries them straight into the liver. And in fact, all of the digestive block of organs, even the spleen, are going to be drained by the hepatic portal vein. And the hepatic portal vein is going to carry the blood into the liver. And sitting next to where the hepatic portal vein comes in is the hepatic artery. The hepatic artery is bringing oxygenated blood from the aorta. The hepatic portal vein is bringing nutrient-rich blood from the digestive tract. And the blood from the branches of these two vessels mixes in the liver sinusoids, which are the capillaries of the liver. Sinusoids have large gaps in their walls. Plasma can flow freely out of the capillaries into the interstitial fluid and come in close contact with the liver cells, or hepatocytes. Hepatocytes are organized into plates which stack into a hexagonal arrangement to form the liver lobules. Each lobule has 6 corners. Each corner has its own branch of the hepatic artery, branch of the hepatic vein, and branch of the bile system, a bile ductule. So the hepatocytes are making bile and sending it to the bile ductules, but they're absorbing oxygen from the hepatic artery and nutrients from the hepatic portal vein. So as the blood flows through the sinusoid from the outer corners to the middle, the hepatocytes are removing nutrients and toxins and adding in plasma proteins and just sort of adjusting the composition of the blood. And the blood then drains into the middle of the lobule into a branch of the venous drainage called the central vein. And all the central veins of each lobule join together, eventually forming a hepatic vein. And there are 3 major hepatic veins that carry blood into the inferior vena cava as it passes through the posterior surface of the liver. Now, be sure you keep straight the hepatic portal vein and the hepatic veins. Hepatic portal vein brings blood into the bottom side, the inferior surface of the liver, carrying nutrients from the digestive tract. The 3 hepatic veins leave more or less from the top of the liver. They're embedded in the surface of the liver, and they dump into the inferior vena cava. Hepatocytes absorb almost all of the nutrients in the blood, so they get first dibs on the glucose, the amino acids. They're going to remove all the galactose, all the fructose, most of the fat-soluble vitamins, the A, D, E, and K, any toxins including alcohol, mushroom toxin, fungal toxins, drugs that you've swallowed. All of that is going to be absorbed into the liver cells, and they're going to process the toxins and store the nutrients and produce the plasma proteins, and they're taking stuff out and they're putting stuff in. They're very busy, metabolically very busy little cells, and we'll talk about the metabolism of the liver cells in our next chapter. Okay, so we've taken the monosaccharides and the amino acids all the way from being digested in the jejunum, absorbed through the intestinal epithelium, carried through the blood to the hepatic portal vein, all the way to the liver cells. Let's go back and pick up with lipid digestion and absorption, because they follow a slightly different path. Lipids, remember, are hydrophobic, and they have to be mixed with the bile salts. That's emulsification. And the enzyme pancreatic lipase can get into the little lipid droplets and digest the triglycerides down to monoglycerides and fatty acids. And when we're through with all that, we have the tiniest little lipid particles in the lumen of the intestine, and these are called micelles. and they're ready for absorption. So as this little lipid droplet called a micelle brushes up against the microvilli, the lipids in them almost instantly diffuse into the cells, leaving the bile salts behind. Those bile salts can mix with more lipids and create new micelles. Eventually, when they get into the ileum, you actually have receptors that pick up the bile salts there. They're going to be reabsorbed, sent back to the liver, and do it all again. Now, meanwhile, in the intestinal epithelial cells, those lipids have to be repackaged. We broke the triglycerides down to monoglycerides and fatty acids just so that we could absorb them. Now that they've been absorbed, we're going to send them to the smooth ER of these cells, repackage them into triglycerides, take all the other lipids, the phospholipids, the cholesterol, the lipid-soluble vitamins, package them all up in the Golgi with a protein coat, and that makes it a lipoprotein. And these lipoproteins are called chylomicrons. And the Golgi packages them into secretory vesicles, which by exocytosis are going to just dump them into the interstitial fluid at the base of the epithelial cells. Well, they're too big to get into the blood, so instead they enter the lacteal, the lymph capillary. Remember that lymph capillaries have loose walls, loose flap-like, valve-like endothelial cells to let big things in. So the chylomicrons get into the lymph flow, and lymph from the intestine drains into the thoracic duct. And the thoracic duct, you remember, parallels the aorta, carries the lymph up, makes a hairpin turn, and dumps into the left subclavian vein. So at this point, the chylomicrons are dumped right into the blood. Chylomicrons, because they are fairly large particles, will scatter light, giving the plasma a milky appearance. So plasma of someone who has fasted will be straw yellow in color, but plasma of someone who has eaten a bunch of fat is going to be milky in appearance. And this is the reason that they tell you that you should fast before you have blood work done, because sometimes those chylomicrons interfere with the normal blood test that you're trying to have carried out. Okay, so we have the chylomicrons in the blood. Now the blood goes everywhere. From the left subclavian vein, you know it's going to make its way back to the right side of the heart, out to the lungs, back to the aorta, and then from the aorta out through the whole body. Well, that's fine, because out in the body, the blood vessels actually contain an enzyme which can pull the fat right out of those chylomicrons, and the fats are going to be stored in the adipose cells that are scattered throughout your body. The remnant of the chylomicron is going to have the cholesterol, the fat-soluble vitamins, the proteins, and those chylomicron remnants eventually are going to, just through the circulation, are going to get to the liver, and liver cells are going to remove chylomicrons by phagocytosis. So they can stay in the blood for probably hours after a meal. Now, once they've made their way to the liver cells, the liver takes the cholesterol, it takes the fat-soluble vitamins, it either uses it or stores it. It repackages the lipids, including cholesterol and phospholipids, into a second kind of lipoprotein to send it out to feed the body cells. So the chylomicrons deliver fat to the adipose cells, and what's left of the chylomicron goes to the liver, and then the liver makes a new lipoprotein called a VLDL, and VLDL has fat, cholesterol, phospholipid, and these VLDLs go out into the blood. And again, the fats can be removed by the fat cells for storage, leaving behind what's called an LDL. And the LDL is delivering mostly cholesterol and phospholipids and some protein to your body cells. LDLs are actually picked up by phagocytosis by many different kinds of cells in the body. They digest the LDL. They take the phospholipids and make membrane out of it, and the same with cholesterol. They break down the protein of this lipoprotein to have amino acids for their own use, and any cholesterol left over is going to diffuse out of the cell and re-enter the blood. Now, if you have too much cholesterol loose in your blood, you can actually aggravate a condition called atherosclerosis, in which excess cholesterol is deposited in irritated spots in your vessel walls. And this is why LDL levels are supposed to be kept below a certain amount to discourage the deposit of cholesterol in your vessels. Liver cells, though, also manufacture another kind of lipoprotein called HDL. And HDL also circulates through the blood, and when it encounters excess cholesterol, it picks it up. So if you have enough HDL, you're picking up the excess cholesterol, returning it to the liver, and remember the liver is just going to use it, repackage it, make bile salts out of it, whatever. But the HDL removes excess cholesterol from the blood, and that helps maintain the health of your vessels. So you need a high HDL and a low LDL level to maintain your normal cardiovascular health. Luckily, you can raise HDL and lower LDL levels with good diet and exercise. Okay, so we've removed the amino acids, we've removed the sugars, we've removed the fats now, and what's left by the time you get to the colon is waste products that you don't need, don't want, can't absorb, including bilirubin, which you deliberately put there from the liver, water, and salts. And fecal material, as it's processed through the colon, one of the main things you do is remove the ions, the salts, and then by osmosis the water follows it. And one of the principal ions is bicarbonate. And bicarbonate is going to be an important player in acid-base balance. So we'll see that anything that disturbs the absorption of bicarbonate is going to disturb your acid-base balance. Okay, if you absorb too much water from the fecal material, then you have constipation. The material can't move through the colon very well unless it has a certain amount of volume or bulk. And so fiber in the diet is essential. There are two kinds of fiber, soluble and insoluble. Soluble fiber, for example, is pectin, which we use to make jelly out of. It's in very high concentration in fruits like apples, which is why apples are so good for you. Insoluble fiber is cellulose, so you get that from eating vegetables. There's insoluble fiber in fruit and in grains. So if you're eating whole grain breads, you're getting plenty of fiber. The more fiber, the more bulk or volume. The more you stretch the colon walls, the more strongly the walls are going to contract if they're stretched. And this helps propel fecal material through the tract, and it also apparently helps abrade the surface, removing old epithelial cells. And we think this is one of the reasons that a diet high in red meat and potatoes, which is very low in fiber, is correlated with a higher risk of colon cancer. Now, remember those coliform bacteria. E. coli is named that because it's found in the colon. It's a fecal bacterium. Normally, coliform bacteria are non-inflammatory. They live in your gut. They colonized your gut probably when you were born or shortly after birth. They live off the organic waste and food that we can't eat, and that includes certain plant sugars we can't digest, which is why beans are famous for causing flatulence, because they're full of these little sugars that the human digestive system just simply can't handle. So they stay in the fecal material, and bacteria just love them. So anything you eat and don't digest can make its way to these bacteria. And when they digest it, they produce the notoriously odorous and gaseous end products. Another example of bacterial digestion is associated with lactose intolerance. Lactose, remember, is milk sugar. And we have an enzyme called lactase to digest it, but depending on your background, you may or may not continue to produce lactase because milk is associated with the dietary needs of children. Adults don't really need to produce lactase. And if you fail to produce it, but you continue to eat dairy products that have lactose in them, the lactose can be broken down by the bacteria, causing bloating and gas or diarrhea. Remember that these bacteria do us some good. They produce some of the B vitamins and vitamin K, which we absorb from the colon. And they also help to eliminate, by crowding out, outcompeting bacteria that we don't want. Pathogenic bacteria, which can try to invade the intestinal lining, cause inflammation and diarrhea and pain. And this explains why if you're on a broad-spectrum antibiotic, you tend to have digestive upset, because an antibiotic that you may be taking for a urinary tract infection or an ear infection can also kill the bacteria of the colon, and you have to replace them. And we actually actively replace them these days with probiotics, such as yogurts with active cultures. Okay, we're going to talk about regulation of digestion and appetite. we'll start with regulation of digestion. These hormones and nerve reflexes are pretty well understood. So as you learn them, try to separate them into categories. The nerve regulation is through the autonomic nervous system. Remember that the parasympathetic division, whose principal nerves are the vagus nerves, are going to stimulate the stomach and intestine to secrete and to contract, and basically that's going to promote digestive activity. The sympathetic nervous system tends to shut down digestive activity, but the main way that it affects digestive activity is that it vasoconstricts the blood supply to the digestive system when you're physically active and you need that blood to go to your skeletal muscles, and that's why you're not supposed to go swimming right after you eat. There are also these local reflexes or enteric reflexes that don't involve the central nervous system at all. So between the local reflexes and the vagal reflexes, when you put more food into your stomach, that kind of signals the body to hurry up and get rid of what it's already eaten, and it moves things along faster. So putting food in the stomach triggers what we call a gastroileal reflex and a gastrocolic reflex. The ileum has a strong peristaltic wave, just kind of sweeping everything into the colon, and the colon has a strong peristaltic wave sweeping everything into the rectum. And this, of course, is what prompts a defecation reflex, which helps explain why immediately after a meal with a good full belly, particularly infants and children are going to have a bowel movement almost immediately after they eat. Now, the hormones are going to be in two categories. Hormones that regulate appetite, which we're just going to learn a little bit about. We don't understand completely how they work. And the hormones that regulate digestion. So the hormones that regulate appetite obviously are going to fall into two categories: hormones that make you hungry or hormones that make you feel full. And feeling full means you're sated or satiated, and we call that the satiety center. So you have a hunger center and a satiety center. They're in the hypothalamus. And these hormones have a very complex interplay, and we don't know what every hormone is, and we don't know what every hormone does, but we know a couple of hormones pretty well. Ghrelin is a hormone secreted by the stomach. It's secreted by the fundus of the stomach when it's been empty for a while, and it makes you hungry. Now, we know ghrelin does other things besides that. We know there are ghrelin receptors on cells throughout the body. It's a very complicated story. So you can't just say, well, if I want to go on a diet, I just need to block ghrelin, because it has other functions. Besides that, you'd be messing with your body's homeostasis. The opposite hormone that we know the most about is leptin. And when you've eaten a good meal and you've got all those nice lipoproteins circulating to your fat cells secrete leptin when they're full. They've had all the fat they need. They don't need you to eat again. So you feel full. Now, you might think that people that are obese just need to block ghrelin or get somehow an artificial supplement of leptin so that they're not hungry. But in fact, the problem, at least in some people, is that they're not responding normally to those signals. So again, it's not as simple as we think. There are other hormones we know from the small intestine that also influence appetite and satiety, and I feel like we just don't know enough about it to bother with anything more than the simplistic information I've already given you. Hormones that regulate digestion are much better understood, but even then we don't know everything. For example, some of the hormones that we know regulate digestion also are going to have a role to play in stimulating appetite. So the jury's out on it. So the story is unknown on all of that. So we'll focus just on what we do know. We regulate digestive activity through a combination of things going on in your head and things going on in the tract itself. Things going on in your head we call the cephalic regulation. So cephalic regulation of salivation, which we've already talked about. If you smell chocolate chip cookies baking, you're going to start to salivate. At the same time, those same stimuli are going to stimulate the stomach to start secreting stomach acid and to start muscle contraction. So that's the cephalic phase. Just thinking about eating stimulates the stomach. And of course that's parasympathetic impulses along the vagus nerves to the stomach. Once food enters the stomach, it stretches the stomach And even the presence of certain chemicals in the stomach, like proteins, will stimulate the secretion of the hormone gastrin. Now remember, this is a hormone, so it's not being secreted into the lumen, it's being secreted into the blood. And it has to circulate from the stomach cells back to the stomach cells, which seems kind of odd. But being a hormone, that's the way it's going to work. And so gastrin is a hormone from the stomach that stimulates the stomach. So that's the gastric phase. As the stomach is stimulated, pyloric peristaltic waves will push small amounts of chyme into the duodenum. And now we're in the intestinal phase. In the duodenum, when the pH drops because acidic chyme just came in, it triggers the secretion of several hormones which all inhibit the stomach, slow down stomach activity. Let's process the little bit of chyme we have, let it move on, and then we'll re-stimulate the stomach. So the intestine, when the pH is low, secretes hormones that inhibit the stomach, and as that acidic chyme is buffered by bile and pancreatic juice and the pH rises and that fluid moves on into the jejunum, that stimulates the secretion of gastrin from the intestinal cells. So you have a gastric inhibitory peptide to slow down the stomach when you have acidic secretions in the duodenum, and then you secrete gastrin to signal the stomach that you're ready for more. There are two other hormones that are fairly well understood, CCK, or cholecystokinin, and secretin. And these do inhibit the stomach, but they have more important things to do. They stimulate the liver, the gallbladder, and the pancreas. And the role of each hormone is different, so make sure you keep them straight. Secretin stimulates the liver to produce bile salts and the pancreas to secrete bicarbonate. CCK stimulates not the liver, but the gallbladder to contract and expel bile that's already been made into the duodenum. And CCK also stimulates the pancreas to secrete that should say enzymes, not hormones. And as bile and pancreatic juice raise the pH, then the duodenum is going to secrete gastrin, which triggers another peristaltic wave. I'm going to post this as is, even though I've seen at least 4 mistakes in it. I will try to fix the mistakes in the posted copy of this review sheet, but it takes me so long to create these videos that I will not be correcting the video for y'all this semester. If you have any questions of any kind, just let me know, and I'll see you back here for the next chapter. Bye.